intro. So I'm just making this up so I can copy and paste the link and do my little brief lecture thing. There we go to the user dashboard, and then we hit login. Then we go down to LEC SP19, and then we hit plus in the video section, and then we hit external URL. We paste the URL we just grabbed, and then we hit chapter 20, section 1. That's all we're doing today. All I'm doing this morning. All right, I'm going to add and hit publish. Whoops, not there. I am over here, and I am hitting on the Logitech webcam. Yay, my hair's still wet, so that looks fun. All right, so where am I? We're looking at chapter 20, laboratory analysis of the immune response. I'm only going to look at section 1 this morning because, and I'm broadcast early because I'm going to go play chaperone for a science center field trip. So where my They Might Be Giant Science is Real shirt. Wait, I can do that. You know, anyway, I don't have to tell you all that. So anyway, I'm just going to look at the beginning part. This is going to be fairly short, which is probably more appropriate for this format. And that camera angle looks bad, so I need to double check this. Yeah, it's a little bit. Go up a little bit. So after finals, I can bring all the rest of my stuff home, and I can look at a better setup where I can use the green screen. As for no real reason, I'm going to continue finishing that book out. Yeah, which means I have to work on the acoustic because I just got lucky in Chris's old office. The acoustics were kind of okay. And this office is kind of okay too, but my wife is doing a lot of work from home in her job, so this is kind of her space now. All right, so anyway, chapter 20, laboratory analysis of the immune response. We're looking today at section 1, polyclonal and monoclonal antibody production. And then the rest of it we will go through on Wednesday. And then the last disease chapter are going to be digestive. I think that digestive system is all we're really going to get through. And then that'll be it. The so-called dead week. The only thing we'll do then is go on the tour of the wastewater treatment plant, and we'll call it a course. All right, so polyclonal and monoclonal antibody production. So laboratory testing has come a lot further than our microbiology laboratory exercises may seem to indicate. We do pretty old school microbiology there for multiple reasons, but for one is that even the test strips we used to use, those are all fairly expensive items, and you're worth it. Our budget's not quite there. Like Lab on a Chip, this is from Columbia University. This will allow a single drop of blood to be tested for 15 different infectious diseases simultaneously. That includes HIV, syphilis, and you have results in minutes. Now, there's a documentary, I forgot her name and the name of her company, but there's a documentary about that that went really, really wrong for a profit venture there. I hear it's good, I think. Okay, anyway, so the blood's pulled through the tiny capillaries where the patient's antibodies mix with reagents in the reaction chambers, and then there's a chip reader attached to a cell phone that analyzes the results and sends them to the patient healthcare provider. Currently, it's being field tested. So the chip readers will sell for about $100, and the individual chips for $1 is the expectation. That's pretty cool. Of course, markups and all the other fun stuff. You don't know where that's going to land. It's going to be, they'll charge the individual $500 for the chip, even though their cost was $1. And of course, the qualified technician to read it. In this case would be, anyway, sorry, that's a whole different. Clinical focus, part 1. So a healthcare worker struggling with addiction was caught stealing syringes of painkillers, replacing them with syringes filled with unknown substances. That's unfortunate, I guess, is an understatement there. The hospital fired the employee and had him arrested. 2 patients he had worked with later tested positive for HIV. While there was no proof that the infection originated from the tainted syringes, the hospital's public health physician took immediate steps to determine whether any other patients had been put at risk. Although the worker had been only been employed for a short time, it was determined that he had come into contact with more than 1,300 patients. The hospital decided to contact all these patients and have them tested for HIV. Okay, so polyclonal and monoclonal antibody production. I feel like I got some things mixed up in here, but okay. Compare the method of development, use, and characteristics of monoclonal and polyclonal antibodies. Explain the nature of antibody cross-reactivity and why this is less of a problem with monoclonal antibodies. So the cartoon that we used before, as a reminder, the entire complex of molecules is the antigen, and then the actual binding site is the epitope that binds in the variable region of the antibody. So cross-reactivity is more likely to occur between antibodies and antigens with low affinity. Affinity is the binding strength between the antibody and the epitope, and the avidity is the total strength of all the interactions in an antibody-antigen complex. That makes sense because you're talking about different epitopes interacting with an antibody that, it's more likely to have a low affinity for that binding site if it's a cross-reactivity reaction. All right, so producing polyclonal antibodies. And some terminology here. The antiserum is the whole serum collected from an animal following exposure to an antigen. And it is polyclonal because we're directly collecting whole serum. So there'll be multiple antibodies for multiple epitopes. Remember, each memory B cell is producing one type of antibody for an antibody that will bind to a particular epitope. And you have a bunch of different memory B cells and an immune reaction. So you'll have a whole bunch of different B cells that are producing antibodies that will bind for individual epitopes on a particular antigen. So laboratory animals are injected twice. The second exposure, of course, remember the secondary exposure after an initial sensitization. That's to activate the memory cells for IgG production, induce affinity maturation. And so ANTISERA must be purified so that only antibodies of interest remain. So you inject an antigen into, say, a rabbit. The antigen activates the B cells. The plasma B cells produce polyclonal antibodies that are secreted. And you obtain the antiserum from rabbits containing polyclonal antibodies. That's pretty straightforward. Clinical use of polyclonal ANTISERA in many diagnostic tests to evaluate if a patient is producing particular antibodies, and this can sometimes result in false positive or false negatives. Test sensitivity is the probability of getting a positive result when the patient is infected. That's high sensitivity, means a low false negative probability. And then specificity, the probability of getting a negative result when a patient is not infected. So again, if specificity is high, that means you have a low false positive rate. And cross-reactivity is most often the cause of false positives. But that makes sense. Disadvantages. For some diseases, it may take weeks for enough antibody to be produced to be detected. Can be used for immunocompromised patients, obviously, because antibodies persist long after disease. Can't show if infection is active. ANTISERA can cause reaction by complement activation. Cross-reactivity induces false positives, can induce false positives. So monoclonal antibodies. Takes a lot more work to pull this one off. So again, it's same for producing any kind of um, serum. You introduce the antigen to the animal, but then you have to fuse the polyclonal B cells from the mouse's spleen to myeloma cells. So you're immortalizing specific cells. And then you select and grow only the hybridized cells in culture. So these are hybridomas, is the name for that. But they will be producing only, the desired monoclonal antibody that can be grown in large numbers on a selective medium that is periodically harvested to obtain the desired monoclonal antibodies. So it's obviously a lot more difficult, right? It's going to be a lot more expensive to produce, a longer production time, but you do get large quantities of specific antibodies that can recognize a single epitope. Production is continuous and uniform once the hybridoma is made for monoclonal antibodies. For polyclonal antibodies, it's a lot less expensive, right? And fairly rapid production. You do get large quantities of non-specific antibodies that can recognize multiple epitopes on an antigen, which is an advantage, actually, except for the cross-reactivity chance, right? Yeah, but the different batches will vary in composition because you don't know exactly. So monoclonal antibodies. Seriously? Right. That's for the next section, right? Which I'm not doing today because, wow, okay, this will be more appropriate for an internet-length video actually than the other videos. All right, so clinical uses in humans. When mouse cells source for production, we have to create humanized monoclonal antibodies. And that's because where we genetically engineer the mouse B cell to make human light heavy chains. So I don't know how that's a clinical use. What did I think in there? I don't know. You can use this to treat cancer. Herceptin for breast cancer is an example. Few trials in infectious disease due to expense and very high specificity, which can cause the humanized monoclonal antibody to fail to recognize some pathogen serovars. Antigenic variation, right? Current research on using plants to create humanized antibodies, which of course someone wants to call plantibodies. I don't know. I'm not really feeling plantibodies, I gotta say. And this is for the next section, detecting, well, I guess, oh, yeah, so this is another point I wanted to make in here anyway for monoclonal polyclonal antibodies. So again, your polyclonal ANTISERA, this is showing the same epitope here. This is the same. So these antibodies are binding. But you have, this one, antibodies binding a different epitope on the same antigen as this guy and this guy. So you're having a more dramatic agglutination reaction than is possible with monoclonal antibodies alone, which can only basically bring two antigens together, antigen complexes together with a single antibody. You can't have this multiple lattice. Yeah, so less binding occurs and lattice formation generally does not occur with monoclonal antibodies. It's another consequence. Anyway, that's all I'm going to do for this morning, and we'll go through the rest of it on Wednesday. So there we go. So have a good rest of your day, and I'll see you on Wednesday. And here is my outro music that I don't have.